Today we're going to talk about recombinant DNA. There are some points we're going to cover, but the most important points are: what is recombinant DNA, steps of making a recombinant DNA, and uses and aims of recombinant DNA. So what is recombinant DNA? Recombinant DNA technology is the joining together of DNA molecules from two different species. The recombined DNA molecule is inserted into a host organism to produce new genetic combinations that are of value to science, medicine, agriculture, and industry. Since the focus of all genetics is the gene, the fundamental goal of laboratory geneticists is to isolate, characterize, and manipulate genes. Recombinant DNA technology is based primarily on two other technologies: cloning and DNA sequencing. Cloning is undertaken in order to obtain the clone of one particular gene or DNA sequence of interest. The next step after cloning is to find and isolate the clone among other members of the library. Once a segment of DNA has been cloned, its nucleotide sequence can be determined. Knowledge of the sequence of a DNA segment has many uses. Next are the steps of making recombinant DNA. First step is DNA isolation. DNA is isolated in its pure form, which means they are devoid of other macromolecules. The second step is cutting of DNA. For this step, the restriction enzyme are quite vital. It helps to identify the location where a designated gene is introduced into a vector genome. The said reaction is known as restriction enzyme digestion. The third step is amplifying of DNA. Copies of genes are amplified through PCR or polymerase chain reaction. It's essentially a process to increase a single DNA copy into several copies after the desired gene of interest is cut with the restriction enzymes. Step 4 is joining DNA. The vector and a section of DNA is joined in this step. It's achieved with the help of enzyme DNA ligase. Once the gene is amplified and ligated with the vector, it's inserted into a host cell for further cloning. Many different techniques can be used to introduce the ligated DNA into the cell. Once the ligated DNA is introduced into the host cell, the behavior of a host organism changes, and it becomes a genetically modified cell. The inserted gene in the host cell starts expressing itself in the host cell to synthesize the desired protein. Such proteins that are formed from recombinant DNA are called recombinant proteins. Throughout recombinant DNA techniques, bacteria have been created that are capable of synthesizing human insulin, human growth hormone, hepatitis B vaccine, and other medically useful substances. Recombinant DNA technology also can be used for gene therapy, in which a normal gene is introduced into an individual's genome in order to repair a mutation that causes a genetic disease. The ability to obtain specific DNA clones using recombinant DNA technology has also made it possible to add the DNA of one organism to the genome of others. The added gene is called transgene, which can be passed to the offspring as a new component of genome. The resulting organism carrying the transgene is called transgenic organism or a genetically modified organism, GMO. In this way, a designer organism is made that contains some specific change required for an experiment in basic genetics or for improvement of some commercial strain. Despite the achievement of many breakthroughs in the medical biotechnology in the past 50 years, it's apparent that the discovery of recombinant DNA in 1973 by Herbert Boyer and his colleagues Stanley and Cohen at Stanford University Medical School is the single greatest breakthrough in medical biotechnology. A justification for recombinant DNA technology being regarded as the single greatest breakthrough in medical biotechnology is provided by its impact in the field of medicine, industrial process, and agricultural production. Foremost, the use of recombinant technology has led to the development of new vaccines, therapeutic remedies to various conditions including gene therapy for genetic disorders, development of modern diagnostic procedures, and advances in food production through the use of genetically modified organisms.